I think we're going to go ahead and start as people continue to roll in. So I'd like to welcome everyone to this scope webinar. This week, or for this webinar, we have Dr. Richard Bowtell, the Professor of Physics and Director of the Sir Peter Mansfield Imaging Center at the University of Nottingham. After taking a degree in the natural sciences at Cambridge, Dr. Bowtell joined the university as a PhD student under the supervision of Sir Peter Mansfield in 1984. He has spent much of his research career in Nottingham, serving as the head of the School of Physics and Astronomy from 2008 to 2014. His research is focused on the development of new applications and new techniques and hardware for biological imaging. He is a fellow of the International Society of Magnetic Resonance in Medicine and of the Institute of Physics, and past president of the European Society of Magnetic Resonance in Medicine and Biology. As a little bit of housekeeping, throughout the webinar, if you have questions, please type them into the chat. We will get them at the end. I'll read them and Dr. Bowtell can answer. And with that, Dr. Bowtell.
Thank you very much for the introduction. Thank you to everyone who's joined today to listen to this webinar about magnetic susceptibility effects at high field. So I'm going to talk a bit about the effects of magnetic susceptibility. I guess we know the effects can be very good when we think about things like functional magnetic resonance imaging. They can be bad as well in terms of giving us signal dropout and distortions in some forms of imaging. Can be very ugly if we're not very careful. But I guess I'm going to focus on the sort of use of magnetic susceptibility effects to produce like anatomical information, particularly in the brain. In fact, I'm going to focus pretty much entirely on the brain. And what I ended up doing, it's a kind of mixture of, I guess, slightly didactic talk with hopefully some new results. So I hope that's of interest to people who don't know so much about magnetic susceptibility. Maybe you can learn a little bit about that, and also people who have you lost my slides? Just checking there. I'm having some problems. No. Let me just try that again. Okay, sorry about this. I'm going to have to just restart my slides. Find the place. You can see all my directories. And I have to remember which one it is. I think it's that one. And are you still seeing my screen? If I share that, are you?
We see a security alert.
Yeah, I'll try. I think that was the problem. I hope it's not going to happen again. Can you see that? Is that okay?
Beautiful.
Yeah, sorry about that, everyone. So yeah, this is what I'm hoping to cover, a little bit about what is magnetic susceptibility, talk about how it produces field inhomogeneity, talk about the effects of susceptibility on MR signals, say a bit about quantitative susceptibility mapping, and then talk about what makes the different susceptibility of tissues, and then a bit about the effects of microstructure, particularly iron and myelin. So what is magnetic susceptibility? Well, magnetic susceptibility, we write it as chi. It characterizes the magnetic properties of matter. And essentially, it's the constant of proportionality that links the applied magnetic field to the magnetization. So it's how magnetized something becomes when it's exposed to a magnetic field. The field we're interested generally in NMR and MRI is the B field, which is given by the combination of the effects of the magnetization and the applied field that we usually add on with our magnet. So that's the relationship. We're interested in B because that's what dictates the NMR frequency. So as I'm sure you know, chi can be negative, that corresponds to diamagnetic material, or it can be positive, that could be paramagnetic or ferromagnetic materials. So negative diamagnetic materials tend to sort of exclude the field slightly, whereas paramagnetic or ferromagnetic materials actually draw in the magnetic field, enhance the field. So everything is actually diamagnetic. All materials are diamagnetic, have diamagnetic properties. But I guess we're interested in particularly water and fat and bone when we're thinking about the human body. So we have sort of weak diamagnetism with susceptibility values which are of the order of 10 to the minus 5 negative. Paramagnetic materials have positive susceptibility values. Generally, they contain constituents with some kind of net magnetic moment, which produces an alignment with the field, and then the collective effect is to enhance the field. Deoxyhemoglobin is one example of paramagnetic material. Ferromagnetic materials obviously have large susceptibility values that are positive, and they can be magnetized even in the absence of fields. So we generally kind of keep them well away from the magnets that we use in MRI. I hopefully explain a little bit about paramagnetism. Yeah, what underlies diamagnetism? It sort of arises from the fact that the electrical charges within atoms and molecules can be sort of can undergo motion, which will partially shield the interior of a body from an applied magnetic field. So the effect of the magnetic field is to cause a change of the electron motion, and that acts to screen the inside. And that's what underlies the, I guess, chemical shift effects to a large extent, but also bulk susceptibility effects. So essentially, this just scales up with the applied magnetic field, and it's a magnetization which opposes the applied magnetic field. So everything is a little bit diamagnetic, but sometimes that diamagnetism can be swamped by paramagnetic effects. It's worth just saying in passing that this diamagnetic phenomenon can also be anisotropic. So essentially, you can get a different susceptibility dependent on the direction of the applied magnetic field with respect to the sample of interest, but underlying that is the effect of orientation of the field with respect to, I guess, molecules and structures within the sample. So a simple example is a benzene ring, where if you apply a field perpendicular to the ring, you can get a circulation of electric current, of electron current, within the molecule, which gives rise to essentially a slight reduction of the field. So you have negative diamagnetic effect. But if the benzene ring is sort of parallel to the field, you don't get that sort of effect. So there's a change in susceptibility with orientation. And actually, there are quite a lot of biological molecules which can manifest this effect. And we'll come back to this later on, particularly in the context of myelin, where there is an anisotropy, which is linked to the chain structure of the lipid chains in the myelin sheath. Okay, what about the effects of magnetic susceptibility in producing field inhomogeneity? So, as I said, chi is the constant proportionality, tells us how magnetized some stuff gets when we expose it to a magnetic field. So we apply a magnetic field, we get some magnetization in the material, and that produces its own magnetic field, as you can see here. And then that essentially, if we apply the magnetic field in the Z direction, that will dictate the direction of magnetization. But what we need to remember is that if our susceptibilities are small, as I said, for watery stuff, it's sort of typically minus 9/10 parts per million, then the induced fields are actually pretty small. And that means that we only really have to worry about the additional field, which is parallel to the applied field along the Z direction, because this field, which is perpendicular to the applied field, will add in quadrature, and it will make very little difference to the overall magnitude of the field, whereas this field component, delta BZ, will add linearly. So we only really care about the additional field in the Z direction, which is what we have to worry about when we think about the effects of magnetic susceptibilities. Let's take a concrete example. We'll take a sphere of radius r with a given susceptibility chi. Initially, imagine it's surrounded by a vacuum. We apply a magnetic field. it will become magnetized, this sphere of material, and it will produce a field externally, which is the characteristic dipole field. Has this well-known pattern varying as 3 cos squared theta minus 1, where theta is the angle between the field and the vector to the point we're interested in, and r is the distance from the center of the sphere to where we're measuring the field. So the magnetization will depend on the applied magnetic field and the susceptibility. So we can relate the field perturbation to the susceptibility and the applied field. And actually, there's a perturbation in a sort of simple way of looking at this. There is a perturbation also inside the sphere, which is constant in size. However, that perturbation, which you calculate from magnetostatics, sort of assumes the material is completely homogeneous, is completely void of structure. But that turns out that doesn't work if we want to use this sort of analysis to calculate the field seen by the nuclei within molecules, because at that level, we don't have a sort of continuous medium. So what we have to do is use a sphere of Lorentz construction, where essentially we zoom in on the sort of local region where we're thinking our signal is coming from when we're doing some sort of analysis. And then we essentially draw a sphere around the nuclei or the atoms within that sphere. And then we account for everything going on within the sphere. And then outside that, we say it's all a continuum. If we do that, which is nicely described in this paper down at the bottom, then we end up finding out that the perturbation is actually slightly different. There's actually no offset inside the sphere, and then there's the same spatially varying field outside. So the effect of the sphere of Lorentz cancels the effect of the external spherical boundary when we have a spherical region susceptibility. Okay, so then armed with this sort of analysis, this magnetostatic analysis, we can calculate the field perturbation from very simple structures like a sphere, a cylinder that's parallel to the field but very long, a cylinder that's perpendicular to the field that's very long, and we can work out what that field perturbation would look like. And generally, it would correspond to homogeneous field inside the structure and a spatially varying field outside, except when the field is in a long cylinder, there's actually no perturbation outside the cylinder, but there is an offset inside. What about a more general model of the field perturbation produced by a distribution of magnetic susceptibility in tissue, for example, in the brain? How can we work out what that perturbation will be? Well, we can do that by sort of building things up in a simple way. We can say, we'll start with a single magnetic dipole, and we know what the field that produces is. And then we say we've got a distribution of those dipoles. So to work out the field variation from that, we would just convolve the dipole field with the distribution of magnetization. And so, we can build up the field perturbation by summing the effect of all the different dipoles that are present. And we know that the magnetization is proportional to the susceptibility, and it's along the direction of the field. So essentially, we can work out the relative perturbation by just convolving the susceptibility distribution, which we might know under some circumstances, with the dipole field. Well, we can simplify that even further, because you probably remember that if you have a convolution in real space, then if you do a Fourier transform, the convolution turns into a multiplication. So if we Fourier transform the field perturbation and Fourier transform this convoluted susceptibility and dipole field, we end up with the Fourier transform. It's just the product of these two. So if we Fourier transform the susceptibility distribution and Fourier transform multiply that by the Fourier transform of the dipole field, we end up with a simple way of working out what the field perturbation will look like. And we can work out the Fourier transform of the dipolar field in different ways, but it comes out like this, which is actually quite a simple function when we include the effect of the sphere of Lorentz. So essentially, it's a nice relationship in the Fourier domain. I think it had been used in other places in other areas of physics, but we sort of came into use in thinking about susceptibility sort of 20 years ago. Can't believe that. So we can, if we have a sort of known distribution of susceptibility, we can work out the field perturbation it will produce by essentially doing a 3D Fourier transform of the known object function, multiplying by the dipole kernel in the Fourier domain, and then doing an inverse Fourier transform. So yeah, we started with this, we'd end up with something like this, where we see that the field perturbation is projected outside the structure, as we would expect. It's a sum of the field from lots of dipoles distributed in this case to form this iron set of letters. So this is a real example from some time ago. So this is just sort of simulating the field from a set of little cylinders which have been doped with an iron oxide contrast agent. So here's our model, which we generated from a magnitude image. It's a sphere of agar containing little pieces of agar, which within it have this different concentrations of iron oxide material. And we see that by doing the forward model, we can quite nicely simulate this field perturbation in a way that agrees quite well with the measured field perturbation. This is actually the phase variation produced in a gradient echo image, and I'll explain in a minute why that tells us about the field variation. Just as a bit of an aside, I thought I'd just put this in here because, well, I guess this is a scope seminar, and this is one bit where I've used a scope camera, and it sort of uses a forward calculation. So this is some work, recent work by Laura Bortolotti. So the idea is trying to look at the field perturbations produced outside the head and how they change as the head moves. So here's my excuse for putting this in. So if we model the field variation due to head movement, in this case, a sort of head nodding type motion, then this, you can hopefully see the field externally is changing as the head moves, particularly near to, I guess, protuberances from the head near to the nose. So that field, that motion will produce a variation of the external field, which carries information about the motion that's taking place. So potentially we could use the measurement of the external field to say something about the head motion if we had some way of measuring the extracranial fields, the fields outside the head. Well, obviously there is a way of doing that, which is to use a field camera type arrangement. So if we monitor the field at a series of locations, as shown with these little circles, then, in this simulation, you see that there's a pattern of field variation which varies across the probe. So probably carries some information about the motion. So the idea is, can we measure the field variation and then infer what the motion is? So we've done some experiments where we've sort of simultaneously monitored the head motion using an optical camera. So this is the kinetical system, where you essentially have you wear a mouth guard which carries a marker which is tracked by the camera, and then simultaneously we monitor the changes of the extracranial field using a setup, well, in this example, where we have a sort of a quite regular arrangement of the probes. We've also done it where we've just placed the probes inside our 7T scanner between the receiver coil and the transmit coil. So here's some example of these are measurements of the head motion with the optical camera for head shaking, head nodding, wiggling your feet whilst keeping your head still, but that moves your head a little bit, and then trying to keep still. And you see that, obviously bigger motions for these head shaking, head nodding, quite big motion, translation and rotation, and small motions when at rest. And then these are concurrent measurements of the field variation made using this setup with the field camera, where you can see that the field changes do sort of track the motion that's going on. when we get to the rest state, we see that there are some field variations which don't really reflect the motion very well. They relate to movement of the chest in respiration. So that's going to be a confounding effect. So it's a bit of a work in progress, but we do find that for small motions, we can predict reasonably well, predict the motion parameter from the field variations and compare that to the motion parameters measured using the optical camera, get quite a good correlation using a neural network essentially to relate the two and learning from the optical measurements. It's not such a good relationship when we look at large movements. So there's more work to do, but it's sort of an interesting application, I think, of relating field variations to the susceptibility of the head and to the motion. So these, measurements of extracranial fields, they could be used to predict all head motion or at least to flag large movements. Provides a kind of contactless measurement because we don't need line of sight once we don't have the camera in place anymore. But it does require some way of training the system to relate the head movement to the field variation. And we're not doing very well at predicting large movements at the moment. And it's not really yet integrated with the scanner, but hopefully that might be a little bit of interest. Let's now move back to looking at the effects of magnetic susceptibility on NMR signals that we measure in imaging generally. So, we have our simple setup here of a sphere and it's exposed to a magnetic field. We can work out the signal that will be produced by this arrangement by looking at the spin density at each point, and then working out how the signal will evolve at each point, knowing what the frequency offset is as a result of the presence of this piece of material. So if we sort of look at a region and we home in on a little sort of tiny, an isochromat effectively, then in different locations, we're obviously going to have different frequencies because the field is varying with position. And so if we measure the signal from a voxel, then fairly obviously when we sum up the signals from those different regions, they superpose, and as a result of the slightly different frequencies giving rise to slightly different phases, we end up with a decaying oscillating signal. And the decay obviously relates to the rate of transverse relaxation, so R2 star, 1 over T2 star, and the frequency delta omega is the average frequency offset within the voxel in this example. We could also relate that to the frequency distribution within the voxel, where the width of this frequency distribution, at least should it be sort of Lorentzian in form, would give rise to, well, we could relate the width of that distribution to the relaxation rate. And the center of that tells us about the average frequency. Okay, we should remember that obviously in the real world, our voxel is likely to contain a heterogeneous distribution of magnetic susceptibility, as I've sort of described here, which is a simulation. It's a set of spheres within a cubic region, calculate the field variation using the Ford model, and then look at the frequency distribution. So you can see that, that microstructure is going to be quite important in dictating the average frequency and the relaxation rate, and I'll come back to that later. But just sticking with this simple example, then if we make a gradient echo image in the presence of a heterogeneous susceptibility distribution, and we look at the modulus image, then obviously the contrast depends on how much the signal has decayed approximately at the center of the echo. So it depends on the T2 star quite strongly, and that at 7 Tesla gives us pretty nice contrast. But also we can look at the phase of the signal, and that phase will depend on the average field offset within the voxel, which we can relate to the average frequency offset within the voxel. And if we do a little bit of processing, which I'll talk about in a minute, then we also get really beautiful sort of anatomical information from the phase images, which tells us about the distribution of susceptibility. So, simple gradient echo image, as I'm sure you've seen many times, carries some nice information in its phase and in its magnitude. I should of course say, if you've looked on the scanner, that's what the phase image typically looks like, with a reasonably long echo time at 7 Tesla or at other fields, because you end up with wrapping in the phase, because the phase is only defined modulo 2 pi. And so you have a large variation of field, which produces a significant variation of the frequency. The product of that with the TE then could be several multiples of 2 pi, and you end up with these abrupt changes where we classify the field variation in terms of can you just check in, can you still see my slides?
Yes.
Good. Okay, something's gone wrong with my end, but I hope I'll be able to just, yeah, it's all good. So so how do we get rid of this phase variation? Well, there are lots of different ways of doing that. You can sort of just start with the seed region and track along different directions, work out whether there's a 2 pi change, and then sort of effectively reconnects the phase values across that abrupt change. Or you can do a Fourier method to get rid of this effect with some kind of Laplacian filtering. So if you do that and you go from the raw phase to the unwrapped phase, you now see there's a much bigger variation of the phase in radians, because we've gone over several multiples of 2 pi. And then if you high pass filter that phase variation, you end up with something where you can see interesting anatomical structures. So what does that mean? High pass filtering, essentially you're picking out spatial variations which are occurring relatively rapidly. So you take out the large scale variation of the phase and you focus in on the things that are varying quickly. So the place where that's widely used is of course in susceptibility weighted imaging. So essentially you take the high pass filtered phase, you apply some kind of filter function to it to, in this case, darken regions where you have a kind of increased phase, particularly which might happen in vessels with a particular orientation. And then you multiply that into the magnitude image, maybe several times. And then you sort of enhance the contrast, particularly in venous structures, as you can probably see here. And if you take a minimum intensity projection across a slab of the brain, you see, it's really beautiful contrast. You can see the motor strip quite clearly defined there. So we've used this kind of contrast at 7 Tesla in looking at a structure in the deep brain, in the substantia nigra, where there are changes that go on in Parkinson's disease. So Penny Gowland and colleagues sort of used these T2 star weighted images initially at 70 in post-mortem tissue compared with, histology, and showed that there's a region in the substantia nigra which appears bright in normal subjects, but that structure is lost in subjects with Parkinson's disease. So this was sort of assigned as being the nigra zone. I think there's a debate going on as to whether what's seen is actually the nigra zone now. But there's definitely a structure which is present, which you see as a result of this susceptibility contrast. And then this was transferred to 3 Tesla, sometimes being called the swallowtail sign, which is present. So you can sort of see it here maybe, which is present in healthy subjects, but not present in Parkinson's disease subjects. Sorry, I've lost the labeling here, but these are the healthy controls on this side at 3 Tesla, and these are Parkinson's disease subjects where you don't see that swallowtail structure. Okay, a little bit about quantitative susceptibility mapping. So essentially, we've seen there's a quite simple relationship between the frequency offset, the field offset, and the susceptibility distribution, which we can invert, in fact. So we can measure in a gradient echo image with a little bit of processing what the spatial variation of frequency is. Then if we Fourier transform that, we know there's this simple relationship between the Fourier transform of the frequency offset and the susceptibility distribution. And we can invert that relationship to find the susceptibility from a measurement of the frequency variation. So that's what underlies quantitative susceptibility mapping. So sort of looked at it in a simple form. It looks okay, but there are a few problems here which are sort of hidden in this, just looking at this equation. The first is that this function, which we divide by in order to find the susceptibility, it goes to 0 along 2 conical surfaces which lie at the magic angle to the field direction. So we have these cones of doom, which in fact we can deal with not too difficultly. We also have the fact that we don't really know the field variation, the frequency variation everywhere in space. We only know it where we have NMR signals, so we only know it in the brain. And also the field we measure inside the brain, when we measure the phase variation, that depends not only on the field produced by structures within the brain, but also on structures outside the brain, like the body or anything in the magnet or inhomogeneities of the magnetic field. So we need to somehow separate the internal from the external field variation. Well, I'm not going to talk about how that's done, because there's been a huge amount of work in recent years, which I'm definitely not an expert in sort of image processing, computational analysis, and there are a whole load of different toolboxes which allow you to solve this problem in a sort of reasonably straightforward way, which do this processing. We haven't settled on, I guess, a single approach yet, but there's efforts within the field to try and come up with a sort of standardized approach, definitely to acquisition and maybe in the future to processing. So just to show it does work, so this is our phantom again, spherical agar phantom containing little pieces, little cylinders of agar, which have different levels of iron oxide contrast in them. This is one phase measurement after some processing to unwrap and take out the external fields. And then you see after inverting to calculate the susceptibility, you get a pretty good map. This was actually measured by combining images with the object at multiple orientations to the field, which is often sort of viewed as the gold standard way of calculating the susceptibility, sometimes called the Cosmos approach. But obviously it's not very practical for imaging people's heads, but it's at least it's something where you can do it. You kind of overcome a lot of the issues that are present in single orientation measures. This is some more recent data that's colleague in Nottingham, Toby has generated, Toby Adeyemi has generated, and this is using, just for people who are interested, this is the QSM box approach that Giulio Acosta Cabranero came up with. So this is 70 data, quite high resolution, based on 2 echoes, where you see, the kind of beautiful structure of the susceptibility distribution in the brain. So is that going to move on? Yes. Okay, so a couple of areas where we've applied this. So we're sort of looking at how you could do QSM in multi-site studies. So we actually did, as part of the UK 70 network, which I'll come on to describe in a minute, we did a traveling head study where we looked at 10 healthy volunteers, who relatively young, who were scanned on 5 different scanners. So they were scanned on one scanner 5 times and then on 4 other scanners once. All 70 systems, part of the network of 70 scanners in the UK. And then we looked at calculating the susceptibility maps and the variability across, I guess, subjects and systems. So in the UK, it's worth saying as a side, there are actually now 7 Tesla systems, but at the time of this study, there were 5 of them, which were in Cambridge, Cardiff, Glasgow, Oxford and Nottingham. Most of the scanners are Siemens scanners, but our scanner is a Philips Achiever scanner. So what we did, what the postdocs on this project, particularly Will Clark, who led this, was to sort of harmonize the series of different scans across the 2 types of Siemens 70 systems, the Terra and Magnetom, and the Philips Achiever. And one of the sequences that we harmonized was a gradient echo, single echo image, 0.7 millimeter isotropic resolution. And I just mentioned that we've analyzed this data to look at the variability of the susceptibility within the hippocampal subfields. So why would we want to do that? I'm sure you know, the hippocampus, there are two structures which is quite low down in the brain bilaterally, which are very important for memory, and I guess sort of tend to get damaged somewhat in neurodegenerative diseases. There's been a lot of work to automatically segment those structures into their subregions based on good quality T2 weighted and T1 weighted MP2 range often images. So we've been just looking in the 10 subjects, just looking at the home scans, the 5 repeated scans, and looking at the variability of susceptibility within these hippocampal subfields across subjects and across repeats. So the segmentation is done using the ASHES protocol, for those of you who are interested. So just to say that we get pretty reproducible measurements of the volume of the different subfields. So these are the 10 different subjects. Each bar is one subject. These are different subregions of the hippocampus. And here the error bars correspond to the standard deviation across the 5 repeat scans. And then here, this is the susceptibility variation. And then over on this side, we see the average values of susceptibility and volume across these healthy, relatively young subjects. So I guess what we find is that there is variability, but only at the parts per billion level, so sort of 1,000 times smaller than 1 part per million across repeats. And the variability across repeats is less than the variability across subjects in this young group. So I guess it gives some confidence in being able to characterize small changes in the susceptibility that in an individual subject over time. And that's what we like to do now, what we're starting to do. So with Akram Husseini, who's a neurologist in Nottingham, we've been looking at people with, I guess, the start of Alzheimer's disease, and trying to compare the measurements of susceptibility and volume in these subjects compared to healthy controls, age-matched controls. So we certainly see clear differences in volume, and we may be starting to see some differences in the susceptibility, but these are relatively small numbers still, so more work to be done here. Okay, a little bit about the magnetic susceptibility of tissues. So, what causes variation of the magnetic susceptibility of tissues? Well, I guess the thing that dominates is, well, two things are important. I suppose the density of stuff sort of dictates often how diamagnetic the material is. So higher density corresponding to bone or calcification tends to have a more negative susceptibility value than that of water. But fat is slightly less dense and has a susceptibility which is more positive than that of water. But also things like any paramagnetic material, particularly iron, that's present will tend to make the susceptibility more positive and move us towards zero, away from the -9 ppm of water. So when we look at susceptibility maps of someone's brain, then we see, well, I guess there are two things that really dominate in the susceptibility. There's these bright regions in the middle. These are the deep grey matter structures, and that's essentially high iron. And then you have these dark regions, which are, in this case, the optic tracts, I think, if I'm remembering saying it the correct way, where you've sort of got, I guess, coherently oriented nerve fibers, in this case, perpendicular to the field. And it's sort of similar in an R2 star map from the same slice in some ways, in that you've got this elevated R2 star in the iron-rich regions, and also an elevated R2 star in these regions of nerve fiber tracts. So the dominant features, if you compare to an iron-stained brain section, you see the basal ganglia, particularly the globus pallidus, the caudate, and they correspond to high iron concentration, and they appear more paramagnetic, have a positive susceptibility. And then high myelination tends to correspond to a negative susceptibility, more diamagnetic susceptibility. And both of these features, high iron or high myelin tend to increase the R2 star relaxation rate. So what about the effects of microstructure, which also feed into what I've just shown you? So yeah, why might these things matter? Well, so I sort of showed you this relationship between what's going on at different locations inside a voxel, how the frequency distribution can sort of be related to the signal decay and the average frequency of the signal. But I said it's going to be more complicated because we have microstructure within a voxel. So actually now let's look and vary some of these parameters in the simulation and see what things affect the frequency distribution. So this is my model, random spheres with a volume fraction, in this case of 6%, a susceptibility difference of 0.1 ppm from the surroundings, applied field of 7 Tesla. And this is what I get as the frequency distribution, where I've set the average over the volume to 0. So I'm sort of forcing the average to 0 in this example. So if we change the susceptibility of these inclusions, then our frequency distribution broadens, as you can hopefully see. So it depends on the susceptibility parcellation within the voxel. It depends on the volume fraction. So if I increase the volume fraction, then the distribution broadens further. So how much stuff we have in these, how many of these packets of susceptibility we have is also important. It depends on the distribution. So that previously was a sort of random distribution. If we went to a ordered distribution, then you can see that the frequency distribution changes quite a lot. So order is going to change what's going on. And then it's going to depend on the shape of the perturbers. So if I compare randomly distributed spheres to randomly distributed cylinders that are perpendicular to the field, you get a change in the form of the frequency distribution. And if I go to cylinders which are parallel to the field, I get a very different field variation, because if you remember, a cylinder that's parallel to the field, there's no external frequency perturbation. So I just get 2 delta functions for the frequency outside and the frequency inside, which is obviously going to give us a slightly strange, it's a not very natural sort of behavior, but it's sort of indicative of what might happen if we change orientations of ordered structures. And importantly, if we lose the signal from one compartment, then if, for example, our cylinders had low spin density and didn't give us a signal, that completely changes the frequency distribution. That's going to be quite important. The same thing's going to happen for cylinders that are perpendicular to the field. If I lose the signal from inside the cylinders, the distribution changes quite a lot. So there's quite a lot that could affect in microstructure, which could affect the frequency distribution and therefore the signal variation. So let's just finish by looking at the effects that non-heme iron and myelin might have within the context of this sensitivity to microstructure. But just non-heme iron, what does that mean? Well, so the majority of the iron, well, a lot of iron is found in hemoglobin, but we're not thinking about that. We're thinking about the iron storage, which is in ferritin, spherical protein shell, packs in a lot of iron in this very, very hydride form, and that's where the iron is sort of stored in the different brain structures. So it's quite, it's the distribution of the iron, the storage iron in the brain is heterogeneous macroscopically. It's different in different brain regions. And it's also heterogeneous at the microscopic scale. If you look at this nice paper from the Vienna group, Simon Hametner, they sort of stain sections for iron. You can see this sort of parcellation of the iron at quite a fine level in different structures. So there's this important work from the 50s by Halgren and Saranda, who looked at the distribution they looked in post-mortem and measured the iron sort of chemically in different brain regions and found this variation across different tissues. And that's actually quite strongly reflected in susceptibility measurements and R2 star measurements of different fields, as shown by the work from the Gratz group, Christian Landkammer, and also, I think this is the NIH group, Jeff Dunn's group. So macroscopic iron distribution, you can characterize it quite nicely with R2 star and susceptibility measurements. But there's other things going on in that if you look carefully, as they did in this Vienna paper, if you look properly, that there are regions which don't really track the iron concentrators very well, and that's because of the effect of myelin. But there's also maybe also an effect of the distribution of iron as well, the way it's parcellated up. And I think that's an area of quite interesting future research, which I'm not going to say too much about. There's some work from Nick Weisskopf's group in Leipzig looking at very fine, comparing histology and characterization of the iron distribution to measurements in the same sort of, with almost the same level of resolution with MR, which is quite exciting. I guess we've been looking a bit at a more theoretical analysis of when interesting effects might happen. I won't talk about it, but there's something at ISMRM. Just to finish with myelin, sorry, I'm sort of getting towards the end now. So we've seen that, very strong effects of myelin. So why is that? Well, these myelin sheaths, they effectively act as electrical insulation. They're fatty structures, which sort of insulate, as I'm sure you know, insulate the axon. So the high myelin content is what makes the tracks appear diamagnetic. It arises because the lipid chains which make up the myelin sheath, which is sort of shown in here, they're long lipid chains which have an anisotropy of their susceptibility along the chain direction. So you end up with this slightly interesting structure where you have an anisotropy of the susceptibility with the principal axis oriented radially, which gives rise to some quite interesting behavior. So just evidence for that came from this work from Chun-Lei Lee's group, I think when he was in Duke University, where they sort of showed that there was a susceptibility, a measurement, apparent susceptibility variation depending on the angle of the nerve fibers to the direction of the magnetic field, which went away in a genetically modified mouse which had dysmyelination. So it's sort of variation with angle, this apparent susceptibility measured from QSM, no variation when the myelin's gone away. We found similar things in the human brain, as did other people. So if you look at the magnitude signal weighted by R2 star, and you get someone who can vary their head orientation quite a lot inside the scanner, you find there's a difference in R2 star, in regional difference, which changes with the head orientation, essentially low R2 star when the fibers are parallel to the field, and high R2 star, fast relaxation when fibers are perpendicular to the field. And also a similar effect if you measure the frequency, apparent frequency of evolution of the signal at two different echo times, and compare that at short and long echo times, you see a similar sort of effect with this negative apparent frequency when you have fibers that are perpendicular to the field. Well, this was sort of explained using this hollow cylinder fiber model, so sort of simplified model of the nerve fiber, which has that radial anisotropy. And the sort of interesting things about that model are that it gives you a negative field inside the axon as a result of this radial anisotropy of the susceptibility in the myelin sheath. So this is, on the top is the effect of on the left-hand side, effect of isotropic susceptibility, where the field is along the fiber and then the field is perpendicular to the fiber. And this is the effect of the anisotropic susceptibility, which is kind of weird, but actually it's just like, it's behaving like a Halbach magnet. So what we found in work some time ago was that you end up with quite significant sensitivity to the fiber orientation to the field in this frequency difference measure, and also in the rate of decay of the signal. So the rate of decay is fast when the fibers are perpendicular to the field, and the frequency difference effect is quite big. So the reason you get this is essentially that you're losing the signal from the myelin compartment. So if you look at the apparent frequency, if I just go back one, so if you look at this thing over here, then as time goes on, you're losing the signal from the myelin sheath. So you have sort of early on, you measure a little offset for perpendicular fibers, and then later on you measure a strong negative frequency offset, which gives rise to this frequency difference. So the way that has been used is in sort of myelin water mapping. So particularly the group of Jong-Ho Lee have exploited this in improving myelin water measurements using gradient echo techniques, essentially by building into the model the frequency offset and how that might change depending on the fiber orientation. So they find, that they reduce the residuals in the fitting of the signal evolution as a function of echo time in multi-echo gradient echo data sets when the fibers particularly are perpendicular to the field. And that's been quite useful. To say these kind of measurements are not that easy, and it's another place where I guess field monitoring is quite important and could have significant benefits. So I think I'll skip on then to just sum up, just to say, hopefully I've given you I guess it was a bit of a whistle stop tour, sorry, at the end, very whistle stop, but talked a bit about what magnetic susceptibility is, talked about how that generates field inhomogeneity, talked about the effects of magnetic susceptibility on NMR signals, and then talked a bit about why the magnetic susceptibility of tissue varies, dependence on iron and on myelin, talked a bit specifically about the effects of non-heme iron and something about the effects of myelin. So to just acknowledge many people who've contributed to this work, sort of past and present. I guess these people listed here are present people. So particularly should highlight, I guess, Penny, who's been involved in a lot of this work, Penny Gallen, Olivier, Toby, who did the QSM in the hippocampus, and then Laura, who did the motion measurements with the field camera, and Akram, who's the neurologist leading on the work in the hippocampus. So thank you very much for listening, and interested to hear any questions that you might have. Thank you.
Thank you. Just as a reminder, if you have any questions, please type them into the webinar chat. So far, we have 2 questions. The first one is, in the raw phase images, in the fringe between the edges, the pi wraps look almost instantaneous at their interface. How does the filter address that abrupt fringe? And are these fringes from macroscopic effects? Presumably, you don't want to remove the microscopic intravoxel signals.
Yes, very, very good question. I guess the field variation, well, I suppose there are multiple things going on at the edges. I guess there is a rapid field variation. It may be that you have partial voluming effects, which give rise to rapid variation of the phase, which is obviously interesting, but it's quite difficult to, I guess, use that information very well in QSM, although there's work, I suppose, going on to try and improve that. But often in what you do in a lot of the approaches is you do some kind of, you have to do some kind of masking if you're looking at the brain to pick out the brain region, and you may end up eroding sort of difficult voxels on the edge of the brain. So that is an issue. Obviously, you can shorten the echo time down. If you've got short echo time measurements, you can maybe reduce some of those effects. There's work to try and, I guess, use the more of the total field variation and combine the analysis where you're trying to separate external internal fields all in one go. So it's all helping, but it's still an issue, the surface effects. It's a good point.
Thank you. That was from Sina. Ali asks, hi, is there a reference such as a textbook focusing on susceptibility effects on MR physics?
I can't think of a textbook, but maybe that's my ignorance. So there are a lot of good review papers. I guess there was a special issue of NMR in Biomedicine, probably 2016, I think, which had a lot of good summary reviews of the different aspects of QSM. So that's a good place to look. I guess there's a very good review from Yi Wang from a few years ago, and a range of other different reviews. So I would start there. And so, but if anyone's listening who wants to write a textbook, then maybe there's an opportunity.
If you don't mind, I'll throw one in. Mark Hakey has a textbook.
Yes.
Yeah. David asks, can you comment on the pros and cons of using QSM for myelin mapping and quantitation? Can it replace direct detection of myelin?
I think I don't think I would say probably not would be my view, just because of I didn't go into the detail, but in the tracks, the effects I talked about of myelin sort of slightly break the simple QSM model. So I think you have to be a bit careful about, if you use a sort of simple QSM approach, and maybe a little bit careful about what the measurement there exactly tells you. Certainly understanding the frequency difference mapping, which I didn't have time to talk about much, but it was what we've done, where you sort of try and model the evolution of the frequency in terms of the hollow cylinder model, which can include parameters about the nerve fiber, the G ratio, the external, all of those things. It's sort of interesting, but I think it's quite hard, because these are subtle effects, which I think, there are many other sources of field perturbation. If you're trying to measure at long echo times and separate small differences in frequency of compartments, that's not a very easy thing to do. But I think what I'm interested in, I think it's worth looking more into doing that. I think in the myelin water mapping field, I mean, I'm not an expert in that area, but the ability to use gradient echo images to do the fitting, including the effects of the offsets, I think that's interesting and seems to be gaining, as far as I can tell, some traction.
That's good. Elix asks, for the neural net trained to extract motion parameters from the field camera, you showed that it worked better for small movements than large, but you also showed that the field perturbations appear to correlate better with the large movement compared to small. Do you have any ideas about this apparent contradiction?
Well, I think my correlation that I showed was a sort of by eye correlation, I guess. So hopefully I didn't, maybe I overstated that. I mean, they're changing in a similar way, I guess, temporally was all I was saying there. So I think there's the effect of, the problem is the effect of respiration for small movements. But I think what we tried to do is to use, you expect the fields from respiration to be slowly varying. So if you use spherical harmonic decomposition, then if you take out the low order spherical harmonics, you can focus in on the fields that are which are coming from the structures on the face, which tend to be more rapidly varying. So I think you've got a way of removing that modulation that appeared in the time course that I showed for the resting state. So that's hopefully a problem that one can solve. But the lack of correlation with the larger movements is something that I'm not sure how to address that. And I think we probably need to look further. Maybe what you would end up doing is just sort of using a simpler field monitoring or something else to just discard measurements where the variations were too large, I think, which is something that Laura's been looking into.
Sounds good. And then one of my own questions, what do you think about being able to monitor something like iron oxidation state or iron spin state with QSM?
Yeah, it would be great. I'm not sure. I suppose you're probably not going to see free iron. I think what you're seeing is ferritin. I think it's pretty clear. I think maybe I'm wrong, but you're seeing ferritin. So maybe you can say something about the state of the iron in the ferritin. So perhaps that's something, although there's lots of, I'm sure there's lots of squid magnetometry measurements looking at those sort of effects. I think I'm sort of interested in, is there any, what about magnetite in the brain, what in maybe hemosiderin and things like that. So maybe there's something interesting to do, to look at the iron state there, which I don't think has been done, more than just saying, bleeds, they produce huge effects. So I think there's possibly something to do there. But I think probing iron, states is probably, maybe in post-mortem and very controlled measurements with comparison with squid magnetometry, possibly.
That's good, thank you. I'll put out one last call for any questions. No, we are running right at 9 o'clock. Okay, and I think with that, thank you again for a wonderful talk. Really enjoyed it. And thanks to everyone for joining us today. And we look forward to having the next scope webinar. So thank you very much.
Bye.
Bye.